The triple sugar ion test is a microbiological test roughly named for its ability to test microorganisms' ability to ferment sugars and to produce hydrogen sulfide. It is often used in the selective identification of enteric bacteria including Salmonella and Shigella. Composition: the Tsi Slant is a test tube that contains agar, a pH-sensitive dye, 1% lactose, 1% sucrose, 0.1% glucose, as well as sodium thiosulfate and ferrous sulfate or ferrous ammonium sulfate. All of these ingredients are mixed together and allowed to solidify in the test tube at a slanted angle. The slanted shape of this medium provides an array of surfaces that are either exposed to oxygen-containing air in varying degrees or not exposed to air. Tsi agar medium was developed based on Kigler's iron agar, which had been used for the determination of lactose fermentative bacteria, by addition of sucrose to be able to detect sucrose fermentative bacteria also. Interpretation of results: bacteria that ferment any of the three sugars in the medium will produce byproducts. These byproducts are usually acids, which will change the color of the red pH-sensitive dye to a yellow color. Position of the color change distinguishes the acid production associated with glucose fermentation from the acidic byproducts of lactose or sucrose fermentation. Many bacteria that can ferment sugars in the anaerobic butt of the tube are enterobacteria. Some bacteria utilize thiosulfate anion as a terminal electron acceptor, reducing it to sulfide. If this occurs, the newly formed hydrogen sulfide, H2S, reacts with ferrous sulfate in the medium to form ferrous sulfide, which is visible as a black precipitate. Examples of sulfide-producing bacteria include Salmonella, Proteus, Citrobacter and Edwardsella species. The blackening of the medium is almost always observed in the butt of the medium. A bacterium that is a non-lactose fermenter and ferments glucose, initially causes a yellow slant yellow bottom after 8 hours but then converts to a red slant yellow bottom after 24 hours. Whereas if it ferments both lactose and glucose, it results in a yellow tube and remains that way due to the large amount of acid produced in the reaction. Blackening of the bottom due to H2S production may mask the acid reaction in the bottom of the tube. Salmonella entericus eruvativi may result in blackening of the medium at the interface of bottom and the slant. Under anaerobic conditions some bacteria use thiosulfate as an electron acceptor and reduce it to hydrogen gas. This is not very soluble and may accumulate as bubbles along the inoculation track, between the agar and the glass, or in the fluid which accumulates at the bottom of the slant. Hydrogen production may lift the agar from the butt of the tube or fracture the agar. Carbon dioxide, if produced, may not show as bubbles because it is far more soluble in the medium. See also, cystine tryptic agar. References